Yeah, so it's my honor to be able to present today about some of the work that I did during my PhD. So the presentation for today is titled as Extracting Allelic recounts from 250,000 human sequencing runs in the short sequence read archive. So later on, I think it got up to like 1 million in terms of the total sequencing runs that were aggregated. But yeah, for this particular analysis, we'll only focus on the 250, 100,000 human sequencing runs for now. Oh, let me. So before I go into the presentation, let me just talk a little bit about what I did during my PhD at Illumina in case it's of your interest. So during my PhD, I was in the UCSD Bioinformatics and System Biology program, also known as the BSB program. So my PhD advisor is Hannah Carter. I also works closely with Jill Masaraf and Trey Attiker on many different computational fronts for my project. So right now I'm a bioinformatics scientist at Illumina. So recently, I guess one of the noticeable projects would be the COVID-seq. So that one is the first FDA EUA approved coronavirus submission, which allows sequencing up to 3,000 samples per sequencing run on a single NovaSeq machine. So that in theory should help increase the throughput and also give more texture and information towards the coronavirus that we collected from the patient. So instead of just telling whether the person have it or not, when they have it, we can also tell what the variants that they have it to trace to different strains that could potentially yield information regarding like where they got a more aggressive strain or they got a lesser strain. So that could also help triage maybe in the future. So it's a very exciting project that's still ongoing. So I'm also the primary developer for Dragon Metagenomics. So that was more focused on using the FPGA to accelerate the computing to map the microbial reads more effectively. And the other product I also work on would be the array Infinium products at Illumina. So it's like the 450K methylation array and also the GSAV3 genotyping array that are predominantly used by the direct-to-consumer market. So like 23andMe and also Ancestry, they rely on more on the array product actually. So just a quick disclaimer. So this presentation only represents my own personal opinions, but not from Illumina, so that I don't get into any legal trouble. Okay. So let's get back to the presentation on extracting the allelic recounts from SRA. So the problem with the public omic data is that it has very poor findability, accessibility, interoperability, and reusability in general. It's also known as a fair problem. If any one of you is familiar with the big data to knowledge roadmap efforts in general. And the significance of this problem is that data preparation accounts for 80% of the work for data scientists. And if you think about it, if you don't need to do a lot of the data preparation, like right now in bioinformatics, then instead of spending 5 days in working, you can spend 1 day working and spend the rest of the 4 days like enjoying the sunshine in San Diego. I don't know if you can see it, maybe.
Nice.
Yeah, so currently whenever a study get published, they usually have to deposit the raw sequencing data in the SRA. And they also have to deposit the preprocessed data in Geo. And sometimes they offer custom databases like TCGA, GTEx, LMBring, at least, and also the ENCODE consortium. But the issue with the preprocessed data that are deposited in the public database is that they are mostly different, generated from different computational pipelines, different quantification metrics, and different metadata annotation methods, which makes it hard for them to aggregate as a single matrix that you can easily play around with. So as an illustration, you can think of the raw sequencing data in the SRA as like the cow. It's like too big. You cannot really eat it directly, right? And the pre-processed omic matrix from the different studies are kind of like beef jerky. They're too processed, and you cannot really revert it back to a steak, like a gourmet analysis at the bottom. So for the SkyMap, it's really trying to reduce into something that you can buy from the supermarket, like the beef, the raw beef, that is everyone will use. So the first question you might ask is why the sequence read archive? So to give you some context, it's actually 1.4 petabases worth of data, and they're deposited from different studies, like over 20,000 different studies from different institutes. So if you assume it's gonna be 10 cents per million basis, that's actually $1.4 billion of your tax money that went into it. And the other thing to note is that the SRA data is very diverse in biospecimen. So they have cell lines, they have primary tissue data. It's kind of like a library. You can find any book you want. And the other thing is that it's produced by many independent studies. And that's another advantage, where if you want to validate your results using independent batch of data, you can just go to SRA. So the question we ask is whether we can improve the fairness of the SRA data that's growing exponentially right now, as shown on the plot on the right. So the x-axis is by year, and the y-axis is the number of sequencing experiments accumulated in the SRA, and you can see that it's growing exponentially. So the goal of this project is trying to generate a allelic recount matrix covering all the public sequencing data, which is one of the aspects of SkyMap. So the motivation is that the genetic studies usually require huge sample size, which is not usually readily available for some rare diseases. And we also observed that the variant calling is mainly based on the real allelic recounts. So whenever we want to evaluate whether a variant is right or whether it's likely to be real or not, we can usually pop up the IGV view like on the right-hand side, and we simply just count over the number of read that support the alternative allele. So which kind of motivate the solution where we seek to generate a single allelic recount matrix to cover all the sequencing experiments in the SRA. So the data layout is fairly simple. So first, the index will consist of the sequencing experiments ID. As you can see, I don't know if I can annotate it. Let me see. Can you see my mouse? I'm just curious.
Yeah.
You can? Okay, perfect. Yeah, so the allelic recount matrix consists of two parts. The one is the index, which is the sequencing experiments ID that is shown here. And then we get the allelic recounts for each of the chromosome position supporting a particular base with a number of read. So that forms the allelic recounts matrix. So for example, in this particular experiment, ERR 12611, at chromosome 1, position 14727, there's 5 reads supporting A and 41 reads supporting G. So that information should be useful for calling variants. That's the hypothesis. But the first problem we first have to solve is that we need to create a fast bioinformatics pipeline to process such a vast amount of data, which is close to like 250,000 experiments that we down selected from the 300K raw sequencing data from SRA. So we noticed that the bulk of the time is actually spent in sorting the entire Lyman file, and it's very slow. So if you look at the pipeline on the right-hand side, it's very standard pipeline on the left, which involve adaptive streaming, aligning the reads to a reference, aligning and sorting an index using the SAM tools, and then just you do counting the allelic recounts using the BAM recounts tool. But when you look at what's on the right, that's the innovation where we use only a subset of the reference region to help minimize the number of reads that we need to sort at the end, which end up helping to reduce the computational time from years to months. So the targeted reference only consists of a 300K or so variance from dbSNP. It's mainly from clean ball and some other significance criteria. Then I retained the 1,000 base pairs of upstream and downstream of each region so that the region become mappable. And that will be our variant specific human reference genome to restrain the mappable region for bowtie 2. Is that somewhat clear?
Yeah, for me, it's pretty clear.
Okay.
Yeah.
So after we have generated such a vast amount of data, we also need to do a systematic validation on the reprocessed SRA data to make sure all the different data layers that we generated are useful. So there are a couple goals that we want to achieve. The first one will be to first show that our pipeline use results that are comparable to the gold standard pipeline, like using our pipeline to reprocess TCGA data and see whether we can recover the regional results from TCGA. And the second goal that we try to achieve would be to evaluate the effects of sequence duplicate removal in variant identification to see whether the assumption of the necessity of duplication removal based on the start and end sites of duplication is holding up or not. And the third goal that we try to achieve here is to identify variants from the RNA-seq using the allelic recounts. And this point is important because the majority of the sequencing done that are deposited in the SRA are done with RNA-seq as shown in the plot on the right-hand side, as compared to like whole genome sequencing and ChIP-seq and whole exome sequencing. So really we'll try to get at the bulk of the meat, which is the RNA-seq. So the validation data set that we chose is the TCGA low-grade glioma cohorts. It consists of 500 or so samples. The reason why we chose it is because it has a matched RNA-seq and whole exome data, which we can evaluate the variance on for the concordance. And the other signature about the low-grade glioma cohort is because the low-grade glioma cohort contains a IDH1 point mutation, which is very specific to LGG, which can be an obvious control. So for the first goal to show that our pipeline will yield results that are comparable to the gold standard pipeline, I first want to illustrate how we can use Pearson correlation to evaluate the allelic recount concordance to really try to make a case where our faster pipeline doesn't really reduce the quality that much. So here we only use one alignment file for illustration from TCGA. So the allelic recounts, we can see from the right-hand side of the plot, they're correlated regardless of using the targeted reference or the full reference, where the x-axis is the log number of allelic recounts from the full reference, the y-axis from the targeted reference alignment. And in this hex density plot, we can see that all the 300,000 sites that we captured, they have a over 99% correlation, which make the conclusion where the targeted reference doesn't skew the read alignment, which is what we care about. Because if it skew the alignments using the targeted approach for alignment, then that would make the data quality reduce, and that's not great. So when we look across the 500 or so samples from TCGA in the validation cohorts, we can see that the targeted alignments can reproduce allelic recounts confidently across the 500 samples. As shown on the x-axis on the plot on the right-hand side, we can see that the allelic recounts correlation between the targeted reference and the whole genome alignments are all above 95%. So this helps us to draw the conclusion where using the targeted reference can dramatically reduce runtime while approximating the alignment from the full reference. So the second point that we want to evaluate would be the effects of sequence duplicate removal in variant identification. So the rationale for PCR duplicate removal regionally is whenever they have the same start and end sites, we should remove them for sure. But that's actually an assumption that has been challenged recently. For example, nowadays, even for the Illumina product, we try to use the UMI, the unique molecular identifier, to actual accuracy remove the PCR. But in absence of that, it's actually difficult to draw from the just the assumption where they have the same start and end sites that they are from the identical sequences. So as you can see from the right-hand side, which is just one TCG and alignment file. So the x-axis is without duplicate removal for the allelic recounts. And the y-axis is the allelic recounts with duplicates removal. And we can see that the allelic recounts correlates linearly regardless of duplicate removal in regions with low recounts, which makes sense because it's not saturated yet. But once the recounts hit over the read length, then we can see that it starts to saturate and you start removing the reads dubiously. One thing we notice is at the end is basically that the allelic fractions are comparable regardless of duplicate removal for most cases, except the very rare cases where it has extremely high read depth. As shown on the plot on the right, where you can see that on the red bean, where the total read counts at the variant sites have over 1,000 reads, then the percent of variance with deviation in less than in the allelic fraction in less than 0.05 is a lot less. So meaning that there's a lot more deviation in the deduplication, and without the deduplication in the variant sites that with very high read counts. So the final results that we observe is that over 99% of the variants have an allelic fraction difference in less than 0.05, regardless of whether we remove or without the sequence duplicates removed. So based on the limited evidence to support the duplicate removal using the simple method of removing the reads that have the same start and end sites, we decided not to incorporate it in the computational pipeline, which is in theory supposed to reduce the computational time also. So the third point, which is also the important point, is really trying to identify the variance from RNA-seq using allelic recounts. So to make the case, I first want to show that using the simple alternative allelic recounts is sufficient to identify obvious variance in whole exome sequencing. So for the x-axis is the allelic recounts on low-grade glioma hotspot mutation of the IDH1 R132H. So the y-axis is the percentage of whole exome tumor profile from the low-grade glioma cohorts. And here simply counting the alternative allele for that particular spot. And we can see that it already exhibits a bimodal distribution with one mode centering at around 0 and the other mode centering around 30. And that's an important observation because like currently we often use some sophisticated variant caller for variant calling in general. But what this is showing is that just the allelic recounts itself sometimes is sufficient to determine whether the variant exists or not. So based on what we have previously, we were able to define the positive sample and negative sample as a truth. And now we're going to see whether we can recover the IDH1 mutation in RNA-seq also. And again, the TCGA has matched RNA-seq and whole exome data, which allow us to evaluate how well the RNA-seq can recover the whole exome sequencing data. So the ground truth that we define is basically where there are more than 10 alternative reads supporting the alternative allele in the whole exome sequencing. And the prediction score for the variance for generating the AUPRC will be the alternative read count in RNA-seq. And we can see that the number of alternative allele read counts in RNA-seq alone is sufficient to generate a high AUPRC, as shown on the plot on the right-hand side. So when we try to expand that one particular variant example to the 300,000 variants, we can see that RNA-seq actually still have a high AUPRC and recovery variance detected by the whole exome sequencing. As shown in the plot on the right-hand side, where the x-axis are the AUPRC of RNA-seq recovering whole exome variants, and the y-axis is a percentage of the 300,000 variants that we try to capture in dbSNP. And the significance of this result is to suggest that there's a lot of mutational concordance between whole exome and RNA-seq. But that makes sense because the RNA at the end is transcribed from the DNA. So these results suggest that we can catalog the mutations of over 200,000 RNA-seq experiments. And that also has a wider implication in the future where there's a high throughput, there's a lot of high throughput single cell studies, a lot more large scale RNA-seq cohorts that are being generated that will be even showing more usefulness of this approach. So here are a couple example on why the reprocessed data is useful. So the first point I want to make is that the reprocessed data enable current studies by variants. So you can see that on average, 1,000 sequencing experiments can be returned per query variants on the plot on the right-hand side. So in this particular box plot, you can see that for the known germline mutation is averaging around like 1,000 on the y-axis. It's a log 10 scale, so like 10 to the power 3 is 1,000. And that applies to somatic mutation too for the known ones. So you can easily go from a mutation, for example, if I want to get all the studies with TP53 mutated, I can simply go from the allelic recurrence matrix and find the ones that with a supporting alternative allele. So the other usage for using the reprocessed SRA data is that we can use it to identify the unexpected variants. So for example, one study, which is a purely single-cell RNA-seq study from Nature Medicine that was published in 2017, they collected one leukemia patient's single-cell RNA-seq and the cell profiling behavior and its responses during the chemotherapy treatments over a 6 months period. So they collected close to 1,000 single-cell RNA-seq profiles in total for just purely transcriptomic analysis. And what SkyMap does is that it can also further show that they have some variants that they did not measure regionally, but can be captured by using the reprocessed data. And there's another advantage of using the RNA-seq for variant calling. It's very difficult to measure both the DNA, RNA, and single cell as of right now. It's just that the DNA is usually just one copy per cell, but the cell natively make multiple copies of RNA in each cell. And what we can see from the plot from the right-hand side is that the reprocessed data were able to detect an annotated BRAF V600E mutation, which is a very significant oncogenic mutation in general. And we can see that that particular mutation that is in orange has a very high FRET score actually, and a high number of reads per cell to support that the variant is real. And we can see that the percentage of the single cells that have the alternative allele is actually very high, which support that this variant is found using the Allelic recount matrix is actually real. And not only we can just find the existence of the variants, we can find unexpected association with the variants using the reprocessed data too. So on the plot on the right-hand side, we can see that the increased expression of BRAF V600E allele is actually associated with disease progression in the regional study that was not found in the paper, but can be found again with the reprocessed allelic recounts data. So the x-axis are the different treatment time points from diagnosis, 3 months after treatment, and 6 months after treatment. We can see that the alternative allele, which is the orange curve, it actually increased in expression for the allele with V600E mutated, but the reference allele is actually decreasing, which might explain why the disease relapsed for the patient after diagnosis in 6 months. So that was a very useful example for why we should care about data reprocessing in general. It's just that we not only can reuse the data, but we can find unexpected associations that was not discovered in the original publication. So there are a couple of things that we could do in the future to further explore these projects. So one could be doing more trait associations, and one would be associating with different metadata to see what are the drug responses and how they would affect the different allele expression across different cell types. So one question to ponder is whether we, in the future, since we have such a huge amounts of reprocessed data, can we just let the machine to automatically mimic the human researcher to associate the metadata with the omic data automatically? So finally, I do want to acknowledge the lab that for my PhD, it was a great lab, and Hannah is a great PI. So that was fantastic time actually. And I also want to thank you, Trey, not listed here, for the NRMB cluster that make it possible to reprocess such a vast amount of data. Okay. Any question?
Yeah, thank you so much, Brian. I think everyone can just unmute and ask questions if they like. But I can start. So have you? So yeah, there's a lot of potential future direction, right? But last year, Getty's group reported the somatic mutation called from RNA-seq files in GTAX and TCGA normal samples, where they think they are mosaic mutations. Do you see the same things?
They're most likely.
So
I didn't catch the last part, sorry.
yeah, so do you are you aware of like Getty's science paper from last year where they look? All right, so they basically they published a science paper last year looking at newly arising clonal mutations in normal tissues, and the idea is that, you know, because we didn't get DNA sequencing of all these samples, but RNA-seq, like you said, could capture these newly arised genetic DNA variants. So then they kind of use that as evidence to show, first of all, RNA variants correlate well with DNA variants, kind of like what you show. But they also show that their new variants arise in different tissues of different, you know, places, different lineage. And I guess kind of in concordance with what the Sanger studies have reported on newly arising clonal expanding mutations.
Yeah, I think along that line of thought, I guess right now it's because of, I think it mainly is because of single cell RNA-seq that will actually drive this trend. For bulk tissues, I think people always doubt whether RNA-seq can call the variants correctly or not as compared to just amplicon, just whole exome sequencing. But my thought is that because now RNA for single cell, they cannot really do single cell DNA sequencing accuracy anyway. It's very hard to capture the DNA without dropouts. That's the argument. So I think that will make RNA-seq a very attractive target to find the different subclones in different cell population. I think that will be an interesting future direction.
Yeah, I suppose for RNA-seq, actually, you capture more information in some ways, right? So one thought is that so you showed that a targeted alignment actually works pretty much the same as a full alignment. How much computation does that save? And are you suggesting maybe we should just still do targeted alignment in the future? It sounds like it would save a lot because you're looking at
Yeah,
the
it's
side.
like 10x. It's basically a 10x speed up.
Yeah.
Yeah, I mean this actually is kind of the last piece that I have to finish before I graduate. So that was like I couldn't wait. That was kind of one of the major driver actually for me to think about how to speed it up. But it does actually make it a lot easier for analysis too.
Yeah.
Because like you can just throw away the rest of the reads and keep somewhere else and work only in a small subset of reads. It's actually, I think that's kind of like after started working, that's a more common approach nowadays. Instead of hoarding on every single reads.
Right.
But nowadays we try to just focus on more subset of the reads anyway. So I do personally recommend using pocket alignment.
Yeah. Yeah.
Easier to even look at it, evaluate whether the variant is real or not.
Yeah. And it looks like you're working on the SQL sky map. Like, what's the vision of the SQL?
The sequel? Oh, the sequel? Oh, the continuation.
Yeah.
Yeah, so right now I'm trying to just on the side projects, I'm trying to create a transform model that can translate directly from the aggregated data to just the metadata itself. So the Google actually published a paper on attention is all you need, like 2 years ago or 1 year ago. So that one actually outperformed every single other method in language translation. So just to give you some context. And that has been the latest state of the art, agreed by multiple companies. Like Facebook published something similar using attention model. They call it BERT nowadays. It's a bidirectional transformer model. And that model not only can translate accurately, it's also explainable.
Mm.
Where the attention matrix is supposed to show which word is supposed to translate to which word. So the question would be whether we can apply the same idea to translate directly from genomic data onto metadata, which is in human language, without any human intervention at all.
Yeah.
Right now it's like there's so many things we need to manually curate, but if Google figure out a way to translate language without any human intervention at all, like not even the word, not even like trying to process. So usually in the old days for NLP, they have to figure out a way to first process the words and put it in a tree, some kind of like lexical tree, or, I don't know, that encode the meaning and the relationship between different words. But like nowadays, we don't even need that. So the question is whether we can use the transformer model to directly translate the genomic data onto the metadata directly.
Yeah.
And whether the attention matrix that we train from the transformer model will be explainable, like human translation.
Yeah. And that would be great. Then we would officially engineer ourselves out of our jobs. You just need to know the EHR data, and then the sequence will be run by Illumina, and we'll just the AI part.
I know. Then I'll just go somewhere else.
That would be very exciting. Anyone else have any other questions for Ryan? I'm actually a little bit curious. What can you describe in a few sentences what the Dragon metagenomics pipeline is? I never heard of that product.
Oh, so Dragon is a flagship Illumina bioinformatics pipeline.
Oh, it's fine from okay pipeline.
Yeah, so we bought a company called Article like 2 years ago. The selling point is that they were able to process a whole genome sequencing in 30x whole genome sequencing in less than I think it's 15 minutes.
Okay, so you can propose a whole sequence and a microbiome sequence.
Yeah, so it's on instrument actually. So on instrument from the sequencer, it's supposed to speed out the microbial profile.
I see.
So you imagine yourself going on an overseas, you click a button, then it's supposed to give you the metagenomic profile for the sequencing runs.
I
Yeah,
see.
that's kind of what it does.
I see.
With some special thing like FPGA acceleration, that's a big thing. Um, yeah, to make it run faster.